So we close the first part and now we move to the second part. And the second part is a panel discussion. And I invite moderator for today, Dr. Vinod Gunasekharan, who is a head of pediatric hematology oncology and BMT unit at Trichy. So over to Vinod. Kindly introduce your panelist and let's start the panel discussion. Thank you.
Thank you, sir. Can you hear me, sir? And my video is visible?
Yeah.
Yes,
We can
yes,
hear
we
you.
can hear you. Yes.
Thank you, SPCTT, for giving me this opportunity to moderate this wonderful session on gene therapy with all the eminent panelists. I would be proud to actually ask questions for all these panelists. And without wasting much time, I will introduce the panelists. I think all have been introduced. Dr. Suparna Chakrabarti sir needs no introduction. HOD in the Department of BMT Hematology in Dharamshila Narayana Hospital and honorary professor in Amity Institute of Molecular Medicine and Stem Cell Research. And Dr. Sivaprakash Ramalingam is our previous speaker who is working in IIT Kanpur as associate professor. And our next panelist, Dr. Sweta Bansal. She is the HOD and senior consultant in pediatric hematology, oncology, and BMT, working at Sir H.N. Reliance Foundation Hospital, Mumbai. So I think so far we had an interesting session on how gene therapy is being taken from the bench to bedside gradually, and now we will discuss about certain relevant points in our Indian context. First question, I would go ahead with the questions. First to Dr. Sweta Bansal, ma'am. What are the currently EMA, FDA approved diseases where gene therapy has been used, ma'am?
Thank you, Vinod, for asking this question. At the outset, first, I would like to thank the SPCT for conducting this session. Very wonderful session. And Dr. Shivalangam lecture was outstanding, I would say. And it's really heartening to see that how much progress we are doing in this field. Now, coming to the question, currently FDA approved diseases. So we have hematological conditions, certain neuromuscular conditions, and we have inherited retinal conditions, certain cancers are there. When we're talking about the hematological conditions, as we have heard in the previous lecture, we have thalassemia, sickle cell disease, we have hemophilia A and hemophilia B, where there is a approved gene therapy is there. So when we hear this lecture from Dr. Shivalangam, actually a lot of things have become clear, where we are using the lentivirus and where we are using the adenovirus for this applicability of gene therapy. So second important indication where gene therapy has been improved is the neuromuscular disorders, in which we are using in the spinal muscular atrophy, which has been shown by Dr. Trivalangan in his lecture in the start only. And we are also using this in the cerebral leukodystrophy. We are using it in the Duchenne's muscular dystrophy, and we are using in the metachromatic leukodystrophy. In many other indications of neuromuscular disorder, it is being used, but it has not been approved. So there are still phase 1, phase 2 trials which are going on. Similarly, with the immunodeficiency disease also, we don't have any approved FDA therapy. We still have trials which are ongoing. And we have certain approved therapy for inherited retinal conditions and certain cancers like bladder cancer. And we all know about the CAR T cell therapy, which we have been approved for the leukemia as well as lymphoma.
Thank you, ma'am. Next to Dr. Suparno, sir, which other diseases would you think gene therapy might have an impact in the near future, sir? So I think you're muted.
Yeah, thanks for asking. See, we have gone through a wonderful session. I think we should all absorb this rather than sort of deviating from too much from it. Sorry, I'm saying this because it's been a very wholesome session where some very innovative work has been spoken about. I mean, the approach that Siva Prakash talked about is different from what has been done elsewhere. So taking a cue from that, I mean, you can look at any monogenic disorder, okay, where you have a target cell which you can transduce. So whether it's a, I mean, this takes me back to the sort of late 90s where I was working on adenovirus in transplant. And the work was with a gene therapy group who were looking at adenovirus as a vector for transducing cells. And what was found at that time was that although it was excellent in transducing, but there was a very strong immune reaction that was mounted against adenoviruses generally, because there were 51 uh, 51 different serotypes, and 49 of them there is cross reactivity of the neutralizing antibodies. So that was a big problem with adenovirus, and in the late 1990s, early 2000, it was when adenovirus was used as a tool for gene therapy in one of the conditions in a young girl. There was a fatal fulminant liver failure as a part of the immune reaction. So when we are talking of recruiting other diseases for gene therapy, there is a whole host, there is a whole list of diseases. I mean, at the given point of time, with the range of nucleases that Siva Prakash spoke about, starting from talons, ring fingers, or okay, plus minus, but talking of talons to CRISPR-Cas9 in particular, which has made it so much easier to sort of break and join things like a child's play to an extent. You can look at any disease, but the issue is not about the disease itself. The issue is about what is the fallout of the particular break and join that you're going to do. So with that respect, as I said, it can be any monogenic disorder that you can think about outside the standard sickle thal and certain neuromuscular disorders. And if you take the example you have put here for the DMD, I mean, many of you might be aware that uh, the gene editing that was directed towards the dystrophin gene using the AAV virus created a humongous immune response, very similar to what was witnessed with adenovirus itself. So, as again it was said that there are several types of AAV, each targeting having different tissue tropism. So it again depends on that and how much of the immune reaction it will cause. Now, when this immune reaction happened in the DMD gene therapy, people were expecting that it was because of the genetic manipulation itself. It was not. It was the innate immune response. So you start thinking that you have cracked it. But when you think you have cracked it, nature has something for you. Because what we have to think is, and what we have to stop thinking is that we cannot be God at the end of the day. We cannot cure it all. So we have to follow the laws of nature. And you know why CRISPR-Cas9 is successful so much? Because it follows the law of nature, which has been existing since time immemorial. How bacteria have warded off viruses through this mechanism, which we have realized only now. And there are many more in the pipeline. I am pretty sure that if we look into the smaller beings, we'll find how they have survived billions of years in this adverse environment. There is a lot of it. And if we look into it carefully, we'll find a lot of it. But when we try to think of ourselves as, we have a godlike syndrome happening, we'll be ending up with a disasters more than success.
Thank you, sir. Thank you. That was really an eye opener for us. And Dr. Siva Prakash, sir, what are the recent advances in gene therapy strategies for pediatric cancers, especially?
Yes, sir. So for pediatric cancers, only the CD19 targeting for the ALL, BALL has been approved. That is from the Chimera actually. So other than that, there is no approved therapies currently available for the pediatric cancers. So there are some research is going on for the brain tumors. There is a project called STAR project. There are 3 universities, Stanford, Harvard, and the one university from Canada, McCallum, I think. So they are trying to develop a cell and gene therapy, cell-based therapy for these pediatric brain tumors. Okay. So, but currently there is only one approved product for the pediatric cancer, which is the CD19 targeting for the BALL. I think that is from the Chimera. And the Indian product, the next CAR, is also, I think it's in clinical trial, but it is not yet approved for the pediatric, if I am correct.
Sure, thank you, sir. I'm sure we will get more in future. So again, Dr. Suparno, sir, like what are the potential concerns regarding the gene therapy? I think you have touched upon few points. Like, please elaborate on this.
Yes, I mean, I think again, Dr. Siva Prakash alluded to it at the beginning that one of the problems of gene therapy, again, I was sort of a witness to this. I was in the UK at that time when X-linked lymphoproliferative disorders were targeted with gene therapy, and not one, but I think 4 or 5 out of the 11 patients developed myeloid leukemia because of what we call insertional mutagenesis. When we are introducing, because whenever you are introducing a particular sequence, you're introducing it with a promoter region. You need the transcription of that particular genetic element. And that uh, promoter can get inserted into oncogenic region, and from being a very sort of benign situation, it can become a fulminant malignant situation in due course, leading to various sorts of malignancies. Now, in recent times, during the Bluebird trial in sickle, there were 3 and 5 years down the line, there were 2 patients who developed AML and MDS, which resulted in halting the trial for the time being. This was using lenti. Now, lenti is supposed to be, much more benign because these are all replication deficient viruses, mind you. So when the regulation of gene therapy is very, very stringent. You have to document that these viruses are replication silent viruses. Otherwise, you just cannot get it. So these are replication deficient viruses, but even then, the 2 patients developed of sickle cell disease. So what was shown subsequently is that, okay, there was no insertional mutagenesis. So this particular gene therapy product is not responsible for it. Now, the authors concluded that, okay, it is busulfan which caused it. Mind you, one of the big problems of the hemoglobinopathy gene therapy is that you have to myeloablate the existing marrow to get the edited gene stem cells into the system, because otherwise it's not going to happen. The dominant hematopoiesis is through the deficient or the defective stem cells. So you have to ablate those stem cells. And what you're using for ablation is busulfan, which is a very potent alkylating agent, which has got a strong potential to induce secondary malignancies. So it was easily attributed to busulfan. But what is often missed is that many of these patients have pre-existing mutations, potentially oncogenic mutations, which are otherwise silent. Now, this has been discussed by, one of the greats in this field of hematopoietic transplantation, Richard Jones, a few years back, 4 or 5 years back, he discussed this issue. And not many people talk about it. So they say it's either the edited gene product, or it is the busulfan which is responsible. It's neither actually. When you have, so in a normal state, you have a homeostatic hematopoiesis, which means that cells are all in equilibrium. No one is growing more than the other. But when you have myeloablated and put back a certain number of cells in the system, which are only 2 to 3% of the existing cells, which is true for any transplantation also. That when you are transplanting limited number of cells, which I think recently I spoke about it, that when you are transplanting limited number of cells, why do you need more CD34 cells in an autologous transplant? The very reason is this, that the lesser number of cells you give, it's not that it's not going to engraft, it's going to engraft in a very stressful environment, so that they have to regenerate with 1,000 times more effort than it would do otherwise. And this is what would lead to a select clone, which otherwise was silent in a given homeostatic situation, turn into a malignant clone. So this is what one will have to understand, that it's not one or the other. There are other factors in play. So we cannot have a binary situation with gene therapy, that it's either the gene or the conditioning agent which is responsible. And saying that, we have to be very careful in using busulfan in conditions such as hemophilia and others, where there is the disease is not a hematopoietic in nature. So that's saying that. The other aspect is obviously the generating the immune response. And we have seen more than once that introducing such a group of cells with virus edited genes can lead to unexpected immune responses and death. So these are the two things one have to be you know very worry and conscious about. Thank you.
Thank you, sir. It was really interesting information. And coming to Dr. Shweta, ma'am, like what are the barriers for gene therapy in our Indian context?
I think we know the most important thing is that to understand better about the gene therapy and its applications. It is definitely not a simple thing. It has a lot of concerns, the potential concerns, very well enumerated by Dr. Suparna also. And when we are talking about the gene therapy in India, we definitely need it over here. But I think the cost is definitely prohibitive. As we say about the SMA, which has been used for the Jaipur child, that gene therapy costed around 17 crores. And for the other neuromuscular dystrophies, it is like ranging from 30 to 35 CR. And for hemophilia, it ranges from 1 to 3 CR. So definitely cost is an issue over here when we are talking about the gene therapy. And considering the Indian potential, I think we can develop our own indigenous techniques, which Dr. Shiva Prakash is already working on. And I think that is the way to go on. And next is, I think the most important limitation is the lack of infrastructure. We don't have those kind of a GMP facilities. We don't have that kind of an expertise. We don't have those kind of scientists, and we don't have that kind of an ecosystem where we can actually encourage the youngsters or the young breed to go for these kind of research work, which can actually get translated to the bedside so that we can benefit it to the patients. So definitely we need to develop a lot of bedside. And lastly is about the socioeconomic and the patient mindset, which again we need to create a lot of awareness, because in India any kind of a trial or a clinical study is taken with a lot of pinch of salt, and the parents or the patients, they have a lot of reservations about it. And I think as a medical community, we need to work on that as well.
Thank you. Nicely summarized the points in Indian context. Sir, actually, I think this question is no more relevant, sir. Is there a need for cancer gene therapy in India? Sir, would you like to comment briefly?
No, today we are not talking of this, so let's move on and discuss what is relevant in today's context.
Similarly, for non-cancer disorders, I think this have been discussed. Like the question to Dr. Shiva Prakash, like access, regarding the access to gene therapy in India, do you have anything to say, sir?
Yes, sir. So I think that the gene therapy field is moving very fast in India. So the government is very encouraging to take it more forward. So we know that already one or two products, actually one is the indigenous one, that is the CAR T cells, but that is targeting the CD19. But there are other groups, including ours, and Dr. Gaurav and other people also working on the bispecific CAR T cells to enhance the to overcome the antigen loss, to enhance the persistence. So that has a lot of scope for the ALL and DLBCL patients, leukemia patients. And there are multiple groups who are working actively on cell and gene therapy in India, but some of the groups, though they are actively working, they are not getting recognized actually. So the most of the money goes into some of the groups, but not all the groups. So but the government is very encouraging. There are multiple missions. Say example, National Pharma Mission. There is a BIROC grants are available. and there is one more called Bio E3 from DBT, Department of Biotechnology, manufacturing, making this GMP facility and other things. So this is all from the government initiative and the regulatory development. So now the SEDESCO is also developing guidelines to fast track these approvals, cell and gene therapy. So I am also in that committee. So soon we will have the Indian version of the cell and gene therapy guidelines to fast track to get the approvals for these cell and gene therapy products for both genetic disorders as well as the malignancies and other things. So then the infrastructure, the government is also pushing because we need to have special infrastructure, CGMP facility. That is the major, and then the trained manpower. These are all not done by regular scientists, right? So this has to have a well-trained, we need to have a well-trained manpower. So those things currently little lagging, but we will catch up maybe in another year or two. So these are the things, but I think the Indian scenario is going much faster than the expected. So we will have a couple of products, the cell and gene therapy products, maybe in next couple of years.
Thank you, Dr. Shiva. Like Dr. Suparna, sir, any industrial perspectives? Like,
see, I mean, I'll take it from where Dr. Swetha Bansal and Dr. Shiva Prakash left. This is something I felt very strongly about, you know. I mean, I'll start with the manpower thing. I mean, anyone who does a PhD in the country, their first goal is to go overseas for a postdoc. No one talks of doing a postdoc in the country. Why can we not get people interested to do postdoc in our own country? I mean, if, you know, the sort of work Dr. Siva Prakash is doing, I mean, people who are interested in that sort of work should be coming to his lab. I mean, can we accommodate people in his lab in the same way a postdoc in US would do? Do we have the funds to accommodate more people? So that's the question we need to answer. I mean, a PhD has to be paid well enough. A PhD is not given enough respect in our country. So this is the elephant in the room which we have to address. Until and unless we respect our scientists, we do not relegate them to the position of technicians. Until and unless we allow them to think independently, think boldly, think in a way that will make a difference, even if it's a mistake, even if they make a mistake, even if money goes to the drain. Unless you are allowed to make mistakes, you are not going to develop something. So where is that scope in our country? So it's not enough to say the government is doing, the government has to do a lot more than that. So first step is to see, it's the same story. I mean, you are producing tons and tons of doctors today, lots of specialists. How many of them are going to the villages? What is the purpose of having 100 times more doctor than what we had 2 years back if 80% of the country is not served? I mean, we have brilliant scientists at a senior level, but we are not being able to interest the young scientists. Firstly, we have to give them topics of interest to do the PhD where they use their mind, not just do it mechanically for the boss. Just complete it somehow, get 2 papers out and get a PhD and walk away. So we have to change the way we think about science overall. You know, then only we'll come to a state where we can say that, okay, we are going to be a developed country in this respect, and we are going to compete with US or Europe. And second thing is what we talked about, infrastructure. Infrastructure is not a problem in our country. You know, government is building roads and railways like anything. So making GMP labs is not the problem. The problem are the consumables. We need to produce reagents in our own country. We need to produce equipments in our own country. We need to crack that. Then only we can talk about going at head to head with others. We have people like, you know, Siva Prakash and Deva Jyoti and Tanveer and everyone. We have, Rahul Purwar has done a fantastic job. Did you ever think that someone 10 years back would sit in India and produce a product which is made in India, a humanized form of CAR T? They have done it. But where is the follow-up of that? Where is the fallout of that? There has not been a tumbling effect on that in this field. So everyone is working in their silos. We are lacking momentum in this field. We are lacking the translational approach because the clinicians are in their silos, the scientists are in their silos. We have a humongous research institute called THST over here in Faridabad. Where is the translation? The institute is called translational. I don't see translation. So we have to get out of our silos. Now, industry plays a major role in this because government alone cannot do it. Industry has to come forward. And for industry to come forward, we have to shed this whole business of, that doctor industry partnership is bad. This is what has been portrayed by the media and politicians. This has to change. Look at US. I was showing it in some talk a few days back that in each of the publications there are humongous number of the conflict of interest which are shown openly. So unless there is an industry academic partnership, how are you going to take it forward? So that's my take on this.
Fantastic view, sir. Thank you. Definitely things should change, as you have mentioned. I think of the last question to Dr. Shweta ma'am, like regarding the specifically on gene therapy for hemophilia in India in the perspective of certain recent publications.
Yeah. Thank you, Vinod, for asking this question. I think when we see the recent publications, especially the paper from CMC Vellore by Dr. Alok Srivastava, without any doubt, I would say that there is a very bright future for gene therapy in India. If we look at that paper very carefully, I mean, the major difference is that the kind of virus we have used over there in those patients, though it's a data of only 5 patients, but they have used lentivirus. And as we know, for the hemophilia, we have adeno associated virus applicable gene therapies which are proved. But this was the first time when lentivirus has been used, and it has been used in a very elite manner, I would say, very fantastically done. And they have done the transduction of the gene ex vivo, and they have introduced this after doing the autologous transplant in the patient. And all those 5 patients, even in the 5 patients, they divided these patients into 2 groups, that is group 1 and group 2, in which one was an enhancer group and a second was the non-enhancer group. But all of them are doing fantastically. At the time of the publication, they have reported that all these 5 patients did not require any replacement therapy. And considering that this is completely done in India, made in India, it shows a huge potential that a lot of things can be done. The major advantage of these kind of therapy is that when we look at the adeno-associated virus, one is that it can be used only in the patients who are more than 15 years old. So it is not applicable for the pediatric patients. But lentivirus can be used for pediatric patients when the phase trials have been done. Second thing is that considering the seroprevalence of adenovirus related antibodies in the patients, it may not be applicable for majority of the patients in India. But this lentivirus can help in utilization or its applicability in majority of our patients. So with this, I think we have to think the way we need for our population. Our Indian population is quite different. Our needs are different. Our socioeconomic conditions are very different, and definitely we need to work on that. And with these bright minds like Dr. Shiva and many others, I think we have a very bright future. Thank you so much.
Thank you, ma'am. As have been discussed in the last 1.5 hours, the journey of gene therapy, which started in 1989, has seen the up and downs, and now have a bright future. Thank you all the esteemed panelists. And now I hand over the session back to Dr. Satya sir and SPCTT.
Hello, good evening, everyone. Hello. Good evening, Vinod. Am I audible?
Yes, sir. Yes, sir.
Gaurav, your voice is breaking.
Yeah.
Better.
So Gaurav is at
Am I audible?
with a family function. And I am fortunate enough that I'm also out of the country, but I was glued to the talk. Guys, this is a small attempt from the SPECT family for all of us. There are people like Dr. Suparno, Dr. Shiva, and there are clinicians like Dr. Gaurav who has the guts to take it from the clinic to the lab and from lab to the clinic, and they dream. So it is our own country. We have to put in our efforts, and thank you everyone for taking out time and listening to the August speakers and the panelists. And it's a promise we will be back on the last Tuesday of next month. Thank you very much. Have a great evening. Good night. Thank you, sir.
Thank you.
Thank you, everyone. Thank you.
Thank
Thank
you.
you so much.
Thank you
Thank
so
you.
much.